So I would invite Dr. Garud and Dr. Groussin together now for the panel discussion. And we'll try to address some of the questions from the audience, but like Christian has done in his session, I first leave the floor open to you if you have any questions to each other about the talks. And if not, we'll move straight to the audience.
That was a really great talk. What do you think are some of the consequences of this industrialization on long-term patterns of genetic diversity across hosts?
Yeah, so you mean genetic diversity in the microbiome, right?
Correct.
Yeah, so it's actually a very good question that I don't have a clear answer yet, because we do suspect a lot of like the standard population genetics processes such as bottlenecks, reduced genetic diversity to occur and being associated with industrialization, potentially also associated with a lower ecological biodiversity. How much the two of them really interact, genetics and ecology, it's the same thing we've been all passionate about. So I do suspect that, for instance, the decrease in diversity might be associated with the increase in transfers. Obviously, you increase the chance of being in close interaction with different bacteria if you have a lower biodiversity. But beyond diversity on its own, what we do suspect is also a reduction in biomass, right? So this is something we're working on right now to confirm. But to me, like much more so than diversity on its own, biomass is potentially also something very important to trigger an increased probability of transfers between bacteria. So, yeah.
Thank you. Very interesting.
Yeah, and so the question I had was about so I already saw one of your previous talk about this preprint and the super interesting study on gene gain and loss in infant microbiome. So I'm particularly very excited to see this very different pattern of gene loss in between the first month of age and between the first year of age, with like an increased rates of gene loss of gene gain later on. So how do you potentially explain that from an ecological perspective? Do you suspect like that stress very initially drive the loss and then progressive adaptation occurs through regaining genes?
Yeah, we've thought a lot about this question, and it was quite interesting. And I wondered if the higher rates of loss initially were potentially due to loss of plasmids, for example, though we have very weak statistical signal because annotation is so poor. Though there was some signal of it, but just not strong enough to say anything conclusive. But I mean, in my opinion, that seems like a kind of a logical way by which you just have a lot of loss. It's like if you're trying to catch a train and you leave your baggage behind on the platform, that seems like a natural way to lose what you had. But I'm not sure, actually, and I'd be really interested to know if there are other possible mechanisms. But then in terms of gains, a lot is happening. It's not just the genes that are gaining, it's also the species. And we know that gene transfer can occur across species boundaries. And so, and gains can only arise when you have another player in the community to give a gene. So I suspect, the ecological influx of a lot of diversity at the species level helps to promote that gene gain.
Thanks.
All right, let's dive into some of the questions from the audience. And I'll go by the most upvoted questions. So the first most upvoted question is from Dr. Diener here at ISB. It's directed to Dr. Garud. awesome talk. Do you think that there may be ecological reasons driving faster evolution in infants? For instance, higher competition due to less variability in the diet carbon sources? Do the rates of evolution correlate with the introduction of solid foods into the infants' diets?
Great question. So I wonder if we see a little bit of DVD here, where, you know, the community is so simple compared to the adult, and there's so much opportunity to fill niches that these evolutionary rates are faster. However, I think that actually another hypothesis could be that we have you know, the need for these strains that are coming from a very established system to a very foreign system. It's like brand new, and they need to somehow establish themselves. And to do so, they might need genetic adaptations. And so those adaptations may be really crucial for the survival of the microbes that get into the baby. But the other process that I think could be generating these changes, which is besides adaptation, is actually just drift and bottlenecks potentially, just a lot of stochasticity. And so one thing that I didn't get a chance to talk about is we see a lot of rare mutations that are private to the baby rise to high frequency and then drop over time. And they do so relatively quickly. And so that suggests that there's just a lot of settling out of what's going into the microbiome. And eventually, I guess, selection will help equilibrate what's going on. You asked about whether rates of evolution correlate with solid foods, introduction of solid foods into the infant diets. And I think it's a really good question. And we haven't actually looked at any specific trends related to metadata, though I think that would be a next step. One thing that we did notice corresponding to the introduction of solids, which is around 5 or 6 months, is that subspecies diversity really sharply increases for some of the species. And so it seems like a lot of strains suddenly come in at that point in time. And and it seems natural that, you know, you're consuming new foods, maybe those have bacteria and they come in, or maybe you need different strains of bacteria to help digest those different types of foods.
Yeah, I think Dr. Good, one of your slides also showed that there's a gain in genes between the month 1 and year 1 in infants. You actually gain more genes in this, which would be consistent with the introduction of new foods and the requirement for more function in the gut microbiome.
Correct, yeah.
Great, thank you. The next question is for Dr. Groussin. It's from Leonor Garcia Bayana. How does stability, strain, gain, loss, frequency of sweeps compare in non-industrialized microbiomes relative to industrialized, which I think you touched upon in your presentation, but if you'd like to elaborate more on it.
Yeah, so it's actually, so I partially addressed this question in the end of the talk with by looking at gene transfers. So we do see that, you know, strain are potentially less stable or maybe more dynamic in terms of turnovers of gene contents in the industrialized microbiome due to increased frequency of gene transfers. But we are also very interested in and currently running analysis on how actually mutations and the behaviors and the dynamics of mutations also vary between strains of the industrialized versus non-industrialized microbiomes. So that's something we can address for a few of the species we have, for which we now have strains from different geographies, but we also have the metagenomes that we're trying to leverage as well. So that's something that is ongoing, and we hope to be able to, you know, present as soon as possible, obviously. But right now, it's only very preliminary. What we focused on first is gene transfers.
Great, thank you, Dr. Groussin. The next question is for Dr. Garud from Priyanka Bologna here at ISB. And it's a bit of a flip side of the same coin that Christian addressed about the ecological drivers of this evolution, and also now the environmental ones. Mainly, does hospitalization or mode of delivery have any effect on the strain replacements at earlier time points?
Thanks. Again, really interesting questions, and I think that's a really interesting point. For example, the cesarean section results in more evolution or strain turnover because at the species level, we know that the types of species that get in are quite different compared to vaginal births. And so those are really good questions for follow-up analyses. For the time being, we didn't really delve into these questions pertaining to metadata, partly to focus on the overall evolutionary trends observed across babies, just to maximize our power there.
Great, thank you. The next question, actually, I want to ask you, Dr. Groussin, and take the opportunity being the moderator. Some of your findings with IgA were really interesting, especially this increase in industrialized versus non-industrialized microbiomes and fecal IgA. Have you expanded on that analysis, looking at the lifestyle information you collected, either specifically within just industrialized individuals or across your whole cohort? Are there some drivers of IgA, of fecal IgA encoding specific bacteria that you've discovered?
Yeah, that's a great question. So in at the very large coarse grain aggregate level qualify as industrialized individuals in this particular group, there are people with like a variety of different diets and lifestyle and origins. So you have like industrialized urban people from Kuala Lumpur, from Bangkok, from Boston, from Helsinki. So it's not driven by, let's say, a given geography or a given dietary patterns. These people have very different diets still. They still have like different genetic backgrounds. So it might not be directly correlated to this. So it's more about potentially the impact of having an industrialized lifestyle on its own. What are the drivers? What are the factors that explain this is still very unclear. But it doesn't seem to be particularly at first driven by, a very specific US type diet, or it's more about like having an industrialized lifestyle and what does that mean in your daily life, having a very hygienic lifestyle, so all these aspects are clearly connected and potentially impacting the microbiome. And I would say that this in return impacts our immune response to these new microbes that we now have in our gut. But this is just theoretical. Now, this needs to be proven, and what we're also working with animal models to actually see the contribution of the microbes themselves in driving this difference in IgA secretion, and trying to really mechanistically identify why the contribution of the microbiome on its own, the compositions of the overall community, but also the individual strains in driving these differences in IgA.
Great, thank you. Yeah, I was wondering if certain drivers like stress, just the general stress that you see in a certain like.
Absolutely. So it can be stress. It can be increased low-grade inflammation, which increases the level of oxygen, for instance, and the increase in the abundance of oxygen-tolerant bacteria. We know that Bacteroides, for instance, are more tolerant to small amounts of oxygen compared to Prevotella or other types of species. And these guys have also LPS, pro-inflammatory molecules that are sometimes targeted by the immune system. So, all these reasons might be all tightly connected to an industrialized lifestyle. Right?
Super interesting. Dr. Garud, the next question is from Annie Zhang. What's the DNDS of SNVs in infants compared to adults? Is microbiome more likely to be neutrally selected for in early life?
Thank you for asking that question, Annie. Let me see if I can find a slide. For space reasons, I didn't go into the characteristics of these mutations, but I'll take the opportunity to do that if I may. So hold on. I wanted to share one thing, which is we looked at the prevalences of these mutations, and Ben actually showed something like this in his talk. And this is prevalence in the HMP cohort. So when I have something at 0 here, it means it's private to the baby. And when it's something here in this bin, it's present in almost all the adults. And by contrast, if we look at the distribution for adults, sweeps in adults, it actually looks kind of different. We see a bunch of mutations at intermediate frequency in the HMP cohort. So this distribution was really interesting to us. And so we looked at the DNDS. These mutations that are private to infants have an elevated DNDS of 1.3, whereas these mutations here have a depressed DNDS of about 0.4. However, I want to note that the confidence intervals for these do cross 1. So it's not definitive evidence of adaptive evolution. Nonetheless, we do see, and the other thing is that we see a lot of reversions that are belonging to this class. So they may be private to the baby, but eventually go back to the, we see a lot of reversions. So we see something that rises to high frequency and then goes back to a state that is common in the broader population. So for example, here, this G is something that would be part of this distribution, whereas the C would be something that is part of this part of the distribution. So it could be a lot of stochasticity, but it could also be some adaptation. And then coupled with that idea of adaptation being a driving force, we do see some evidence of parallelism in functional classes, including the Sus-C gene, for example, that Zhao and Lieberman picked up on. So hopefully that helps answer your question, Annie.
Right. The next question is from Moritz for Dr. Groussin. It's more of a technical one. Are the globally collected samples all processed and sequenced in the same way, or do they get sequenced in local labs?
Right, so that's a great question. So all the omics data and isolate genome data were generated all together here in Boston at MIT and the Broad Institute with the same standard pipelines and the same and at the same sites to minimize the effect, the risk of batch effects. So this was really important for us also to go and travel to all these different sites to make sure that the samples on their own were processed the same way with the same protocols, because we know how much can this affect, the viability of the strains as well that we biobank, but also the genomes themselves, because we know that they can quickly adapt via gene transfer. But we also, as part of the GMBC work, promoting very strongly capacity building. And so we have also a lot of community level microbiome data that are also generated on site by the different collaborators. And they are actually leading their own studies as well, using the microbiome data that we co-generate together to answer questions specific to their own projects. So for instance, using the microbiome data that we have as a baseline for healthy populations from which they can then compare more clinical cohorts locally by sampling and generating the data the same way as we did together. So it's a long answer to say both.
Yeah. And are sample collection ongoing through the same pipeline?
Yes, so sample collection is actually currently ongoing in Pakistan, in Indonesia, and in Nepal, and should soon start very soon in Argentina.
Great, thank you. Dr. Good, the next question is from Sean Gibbons here at ISB. The gain-loss pattern over time in infants is really interesting. Do you see that genes lost early in infant life are then gained, recovered later in life? For example, during transition to a solid food diet?
That's a really great question. And we looked at that because previous paper by Yasser et al. Did a very interesting analysis where they found that certain genes were important for transmission of strains from moms to infants. And we speculated that maybe there is some signal like that also in our data, indicating the importance of either genes in colonization or, like you suggest, in feeding. But actually, we didn't see very much of that at the gene level, interestingly enough. At the SNP level, we did. We saw SNPs that went to high frequency and then decreased, as I showed in that slide, but not so much at the gene level. So I wonder if the infant is just kind of hungry for genes, and rather than seeing fluctuations of the same gene over time, it's just an overall increase in diversity over time that will generically be useful for the microbiome.
Great, thank you. The next question is from Hamza Amar for Dr. Groussin. And it's one that I find very interesting as well, is can you clarify whether or not it is easier to detect more recent horizontal gene transfer within the human microbiome? If so, are there methods to deduce less recent or even ancient AGT events?
Yeah, so you can answer this question in different ways. So if you look at ecosystems where gene transfers happen a lot, such as the gut microbiome, you are very likely to have more power to detect signals and be confident in the signal in the data. But there is also definitely by using the criteria we use, which is basically similarity and looking at genes that are 100% similar over like very long stretch of DNA being present in different species, you're very confident about the fact that there is no other way that this gene was actually transferred horizontally. Cannot be explained by vertical evolution, right? Which we also showed in the paper. However, if you look at more ancient gene transfers, there you need to account for other potential processes such as vertical transmission, gene duplication, gene loss, to confidently call a transfer, because increased dissimilarity between the gene sequences is explained by long-term evolution, and a lot of different processes, such as the one that I mentioned, can explain this genetic diversity in the genes you're looking at. And so these models are usually called reconciliation models in phylogenetics, and are now being developed and are more and more powerful to actually detect what's a transfer and what's actually vertical transmission. And we actually leveraged such models to look at the transmission of bacterial strains or bacteria in the microbiome within host species. So instead of thinking of the transmission of genes within the tree of hosts or within the tree of species, you can use the same model to calculate the frequency and the probability of transfers of strains within the tree of hosts. Because the strains transmit the same way. They can swap, which is equivalent to a gene transfer. They can be vertically transmitted across generations, which is similar to vertical transmission of genes. And so these models are also very powerful to, in a probabilistic way, to put some numbers and confidence numbers in events that you detect, such as transfer of genes or transmission of strains.
Great, thank you. Dr. Garud, I had a question for you based on some of our own research. Earlier this year, we published a paper looking at 16S data showing that as people get older, on the ASV and genus level, microbiomes become more distinct to each individual. And you touched a little bit upon this loss of strains in older age and between your twins. Is that complemented by just kind of emergence of a single dominant strain in each individual microbiomes? Is that what you've been seeing in the research you've been doing on that?
Oh, wow, what a fascinating question. When we talk about strain loss, perhaps it's a little bit, Well, I don't mean to imply a loss of diversity, actually. What I mean to say is that the strains are replaced with something else.
Mm.
And but we didn't really quantify that loss in diversity over time. It is just possible that it happens, and I think it would be quite interesting to look at that at the strain dynamic level. But it is very possible that whatever is happening at a subspecies level could be contributing to the signal that you pick up.
Yeah, very cool. I think we may have time for one or two more questions. Perhaps the next one for Dr. Groussin from Tammy Lieberman. How does the fraction of IgA coding vary by taxonomy in industrialized and non-industrialized populations? Also, great talk.
Hi, Tammy. Thanks for the question. So the fraction of IgA coating bacteria vary by taxonomy. Okay, I got it. Yeah, so it's actually interesting. So I couldn't see these data, but here there is the underlying effect, obviously, of having very different microbiome compositions between industrialized and non-industrialized. So the taxa themselves might not overlap very well, and you can clearly pick up differences in taxonomy. What I would be more interested in addressing is what are exactly the antigens that are differentially coded between the different host categories, and that may be shared across bacterial taxonomies, which I think is the key aspect here. But we do see some Bacteroides, for instance, ASVs or species that are found in both industrialized and non-industrialized individuals, and that have, like, for instance, a much stronger IgA coating signal or IgA coating index in the industrialized microbiome. So the fact that they increase drastically in abundance might relate to this, might not. It's I think a reasonable hypothesis, but that needs to be proven.
Well, I want to thank you both for really fantastic talks, and everyone can join me in virtual round of applause for our speakers. And this concludes session 2, and we'll come back with session 3 after a short break. Thank you both.
Thank you. Thank you. Bye, Nandita. Bye.
Bye.
In March of 2020, there was this ominous feeling around the world. Everybody was worried about how this was going to turn out. We had no idea how long this was going to last. There was a lot of discussions among myself and other leaders and scientists at Illumina, like, what can we do to help the world get through this pandemic?
Early on, during the first patients that were identified with atypical pneumonia in Wuhan in China, the China CDC connected with Illumina.
Now we have public health institutions coming to us and saying, hey, Illumina, we need your help.
Once we came up with the sort of strategy of how we wanted to do the COVID-seq test, everybody that we were talking with just jumped up, started getting some of these sub-teams to come back. You got to remember, at that time, we were home and there was no clear plan for how you bring people back to the office.
So my son is immunocompromised, and so I was very worried about bringing this home too. It was emotionally very challenging, both on a work side and on a personal side.
There's not many times in your career when you can see something come together like this, and the teams that are formed, and you create a bond when you're responding to something that everybody has a passion and believes in.
In that time, that early part, it was uncertainty, and it did feel good to be at a company that was going to play a fundamental role in fighting the pandemic.
The CDC brought up the idea, could we help them to respond quickly until they could get others up and running? And then here we are a few months later, and everybody wants to understand surveillance. So we had the foresight at the time to create an assay that we knew they would need before they even needed it.
Primarily, our intention was to participate in the testing, but as the pandemic has has gone on, the value of sequencing and understanding the new variants has really become so important.
It was definitely very collaborative, very open, and it was really exciting to see people who were willing to put in the extra time knowing that it was something so critical to the CDC, critical to the United States, critical to the world in terms of tracking variants.
This pandemic has raised awareness about sequencing, about Illumina, and about the role it can play in health and healthcare.
All of you, from manufacturing to support to legal compliance, product development, marketing.
Thank you to everyone who worked on this.
Thank you for stepping up. Thank you for jumping into that crazy project.
Thank you for making sure that Illumina did everything we could to support laboratories and clinicians in fighting the pandemic.
We did this. We did it together.
It's a pleasure to speak today at the ISB Symposium, a place I love and that I've been associated with in various capacities for over 15 years. I'm going to speak today from a new position as CEO of Longevity, which is a division of Thorne Health Tech, and we are sponsoring the conference. So Thorne Health Tech is a company dedicated to personalized scientific wellness, which has been definitely a huge interest of mine beginning in my ISB days and working a lot with Lee Hood on a number of topics related to this. And the mission of this company is to bring the scientific rigor of biotech and pharma to the prevention space and extend the length of time that people can have healthier lives. We have two integrated brands that fit under Thorne Health Tech. One is Thorne, which is a natural product, high-end manufacturer and provider, and Longevity, which is an AI deep science division where we focus on delivering health intelligence behind scientific wellness. So here, what we're most interested in is microbiome for obvious reasons. We have a number of pieces that we've pulled together in this effort. The first is that we do metagenomic sequencing. That is through a test focused primarily on the gut called the Gut Health Test, which is available at thorne.com. We do machine learning based microbiome data analysis. We also work with groups, including via Longevity on clinical trials, design, diving in on details related to that. And then we also have a proprietary nutrition database that we link to via AI to link the testing to personalized recommendations that are also delivered via the program that we run via thorne.com. So we also do trials on the efficacy of the solutions. This is one example. I'll be very brief here. It's published in Precision Clinical Medicine. A lot of this is based around a lot of the science from one of our co-founders, Chris Mason, as well as some of the background that I have in microbiome research from ISB. Here we do clinically backed gut health protocol. In this case, was done for a 30-day trial, which showed that personalized interventions were able to reduce symptom severity scores for individuals with IBS, irritable bowel syndrome. And this was a highly personalized approach in the sense that there were 75 people in the trial and 68 unique combinations of recommendations that were used to achieve those results. One of the big things I wanted to talk about in the few minutes I've got today is around a new sample collection technology that we've developed, which we call the microbiome wipe. This has been, I think, a very gratifying thing to get to the market. But basically what it does is it replaces the way that we all typically do these microbiome collections, which is putting down that paper, pooping on the paper, having to scoop it, put it in a vial. A lot of participants, a lot of people don't love that step the way that us as microbiome researchers perhaps do. But what this does is it allows you to use actually just a wipe as normal. So basically no behavior change. And I'm going to show a little bit of the data behind that and take you through that experience, because we think this is going to be a much better experience for people going through and doing microbiome collection. And that's going to, we hope, increase adoption across the community of more microbiomes, which is good for all of us. So here's the bit of data. And if people want to dive into this later one-on-one, I'm happy to go through it more. But what this shows is that when we did the sequencing with the wipe, we actually got better correlation with a positive control, which is basically a shipped in dried ice gold standard positive control. And what we see is that if we compare this to the current technology, we get in fact better sequencing quality. If we look at the predicted measures like calculated scores or prediction of different metabolites and so forth, we find again that what we see on the wipe is actually a bit better than what we see on current technology. So we think we're getting both better quality in terms of being able to get sequencing and predictions, as well as also having a better customer experience. So participants, as they go through research studies that you may be conducting, there's a very simplified protocol for this then, right? You get some simple instructions. Basically, you wipe as normal, put it in a container, you shake till it's dissolved. It takes about 10 seconds for that to happen. You release saline, which a solution basically that preserves the DNA, throw it in a container, and you send it back. So that's all I have time to talk about in these 5 minutes. But the test is available at thorne.com for anyone who is interested. If you have questions, want to dive in on the science behind this, or are interested in partnering even on research studies, we do that both from the technology as well as computation side for those who need that. Feel free to reach out to me at nprice@longevity.com.